So today I'm going to talk about the arterial supply to the central nervous system in the domestic mammals. Just as a disclaimer, I don't own any of the rights to this information. This all comes from King, the textbook for the anatomy of mammals. I'm strictly doing this for study purposes for boards. So to start with, the arterial supply to the brain. There are 5 pairs of arteries that supply the brain. The first 4 of these arise from the cerebral arterial circle. If I can get this to work. So here's our cerebral arterial circle, also known as the circle of Willis. It's in the ventral surface of the brain. And the arterial circle roughly outlines the hypothalamus with the stalk of the pituitary at its center. And also here's going to be cranial nerve 3, but we'll get to that later. The 5th arises from the basilar artery. The 5 pairs of artery are the rostral cerebral artery, the middle cerebral artery, internal carotid artery, caudal cerebral caudal cerebral artery, rostral cerebellar, and caudal cerebellar. You can see that inflow where I messed up, the internal carotid is actually coming in. I'm doing this raw for anyone who's watching it online, so I apologize for my mess-ups. There are also various smaller arteries which supply the medulla oblongata and the pons. And more information on that is in the De La Junta book. Although there are minor species variations, these vessels occur in mammals generally. The cerebellar arteries are variable in numbers and origin even within the same species. For example, in man and horses, the rostral one may give rise to the basilar artery. The 3 cerebral arteries are remarkably constant in amphibians and higher forms generally. I am a little bit under the weather. I think it's allergies too, so that's not great. The connection across the midline and the rostral end of the circle is inconsistent in the dog and the ruminants. So there are 4 potential arterial channels to the cerebral arterial circle in mammals generally, as shown in this figure. Channel 1 is the internal carotid artery. Channel 2 is the basilar artery. This midline artery is a continuation rostrally of the ventral spinal artery. However, the blood that flows within the ventral spinal and basilar arteries has come from the vertebral artery via segmental spinal arteries. Channel 3 is the maxillary artery. The maxillary artery supplies the arterial circle by its so-called anastomosing ramus, which joins the maxillary artery of the internal carotid artery. Channel 4 is the vertebral artery. The vertebral artery connects the internal carotid artery, and in some species it supplies the arterial circle directly by this route. However, as stated under Channel 2, it may also supply the circle indirectly via the ventral spinal and hence the basilar artery. Because of the anatomy of these 4 arterial channels, the blood which distributes itself over the brain may be internal carotid blood, maxillary blood, or vertebral blood, or a combination of these. So in this diagram, it shows the potential arterial channels. So there's 4, and they're numbered 1 to 4 on the left. 1 is the internal carotid, 2 is the basilar artery, 3 is the anastomosing ramus of the maxillary artery and the internal carotid artery, and 4, the connection of the vertebral artery and the internal carotid artery. This diagram shows the species variations in the sources of arterial blood to the brain. In each figure, the upper diagram shows the distribution of the brain of the internal carotid, vertebral, and maxillary blood in the intact live animals. The lower diagram shows the anatomy which accounts for the distribution based on 4 potential arterial channels to the cerebral arterial circle. Arrows show the direction of flow in the basilar artery. The vertebral occipital anastomosis, or VO, can be disregarded in the intact animal. Channel 1, internal carotid artery. Channel 2, basilar artery. Channel 3, anastomosing ramus from the maxillary artery to the internal carotid artery. Channel 4, connection of the vertebral artery to internal carotid artery. A is dog, man, and other species. Channel 1, the internal carotid artery, and Channel 2, the basilar artery, supply the arterial circle. The basilar artery carries blood to the arterial circle. Neither channel has a rete mirabile. Internal carotid blood reaches all the cerebral hemisphere except its most caudal part. The vertebral blood supplies the remainder of the cerebral hemisphere and all the rest of the brain. In the sheep and the cat is B. Only channel 3, maxillary anastomosing ramus, supplies the arterial circle. It has a rete mirabile. Channel 2, the basilar artery, carries blood away from the arterial circle. Maxillary blood is distributed to all the brain except the caudal part of the medulla oblongata, which is supplied by the vertebral blood. C is the ox. Channel 3, anastomosing ramus, and 4, vertebral arteries both supply the arterial circle. Each has a rete mirabile. Channel 2, basilar artery, carries blood away from the arterial circle. A mixture of maxillary and vertebral blood reaches all parts of the brain. The rete mirabile is a maxillary rete mirabile. Rete mirabile means marvelous network. Occurs on the anastomosing ramus of the maxillary artery in all species in which channel 3 from the maxillary artery is well developed. Thus it occurs in the cat and in the ox and sheep. In the ruminants, but not the cat, it is immersed in the cavernous sinus. The last part of the internal carotid artery passes through the cavernous sinus. The function of the rete mirabile has long been debated. It is thought that it might eliminate pulsation before the blood reaches the brain itself. However, more recent observations indicate that the rete is involved in thermoregulation. The blood in the cavernous sinus comes partly from the nasal mucosa, where evaporative cooling occurs. Consequently, the blood in the cavernous sinus is cooler than in the arterial blood coming direct from the hot core of the body. Presumably, heat exchange occurs between the rete blood and the cavernous blood, so that the rete blood is somewhat cooled before it reaches the brain. In the sheep, differences of about 1 degree Celsius have been recorded between the body core and the blood of the hypothalamus. It has been suggested that the object of this temperature difference is to protect the brain against rising temperatures. A notable example occurs in certain gazelles which possess an intracranial rete mirabile. These animals can tolerate a body temperature of 46.5 degrees Celsius for long periods. When the body temperature is driven up to 46.5 degrees Celsius by exercise, the temperature of the brain remains about 2.9 degrees Celsius lower than the body core. This lower body temperature probably is due to the heat exchange between the rete blood and the cavernous blood. The rete mirabile arises developmentally from many nutrient arterial twigs which form from a terminal part of the internal carotid near the cerebral arterial circle from the maxillary artery and supply the root of the trigeminal nerve. These nutrient twigs in the fifth nerve are present in mammals generally. In the few species in which the proximal part of the internal carotid becomes obliterated, such as in the sheep, the ox, and the cat, these twigs proliferate and those form a distal remnant of the internal carotid anastomosis with those from the maxillary artery. These anastomoses then form a continuous arterial pathway from the maxillary artery to the cerebral arterial circle, complete with basket-like plexus, the rete miriabile. Now we'll go into the superficial arteries of the spinal cord. Main trunks. Although species variations occur, the basic pattern for mammals is founded on 3 longitudinal trunks, as shown in this figure. Dorsolateral arteries. A pair of dorsolateral arteries runs along the dorsolateral aspect of the spinal cord. The arteries are ill-defined and in some mammalian species, including the dog, cannot be identified as distinct vessels. In man, there tend to be 2 of these arteries on each side, one dorsal and one ventral, to the dorsal roots of the spinal nerves. Vertebral spinal artery. A much larger and more important midline ventral spinal artery lies in the ventral fissure of the spinal cord. So for this diagram of the superficial arteries of the spinal cord in a hypothetical mammal, only the ventral spinal artery is consistent and relatively large in mammals generally. The paired dorsolateral arteries, the anastomosing arterial network connecting these ventral spinal arteries, and the arterial ring at the level of each intervertebral foramen are all inconsistent and irregular in disposition depending on species. The superficial arteries are supplied by paired segmental spinal arteries, which enter the vertebral canal as the dorsal root artery and ventral root artery on each side. Anastomosing arteries. A sparse and very irregular network of arteries connects the dorsolateral and ventral spinal arteries. However, these anastomoses are inadequate as alternative pathways. If the vertebral spinal artery is blocked, the spinal cord will nearly always be damaged. At the level of each vertebral foramen, the anastomosing network tends to be reinforced by an irregular and somewhat incomplete arterial ring surrounding the cord. This ring receives the incoming dorsal root and ventral root arteries. Segmental arteries to the spinal cord. At each segment of the body, 2 arteries on the left and 2 on the right supply the spinal root. These are the dorsal root artery and the ventral root artery on each side. These arteries arise from the paired spinal arteries, which in turn spring from paired lumbar arteries of the aorta, from the intercostal arteries, and from the vertebral arteries. In some species, the dorsal and ventral root arteries are erratically absent from some or many segments of the body. Besides interrupting the sensory and motor rootlets of a particular segment, injury of the roots of the spinal nerves can interfere with the arteries and thus cause degeneration within the spinal cord itself. For the deep arteries of the neuroaxis, the general richness of vascularity of the neuroaxis and the brain and spinal cord is a notable feature. Although the brain forms only about 2% of the body weight, it receives about 16% of total cardiac output and accounts for about 20% of the total O2 consumption of the whole body. Motor areas, sensory areas, and associated areas have increasing vascularity in that order. This reflects functional importance of the associated areas of the brain. The phylogenetic age of the region. The ancient parts of the neuroaxis typically are less vascular than the recent parts. Thus, the spinal cord and the rhiencephalon, the later being phylogenetically the oldest part of the brain, are less vascular than the cerebral cortex and cerebellar cortex. An exception to this is the hypothalamus, which has the richest blood supply in the brain, despite being phylogenetically ancient. But this is probably because it depends directly on high vascularity to carry out some of the functions, such as thermoregulation. The deep arteries of the spinal cord. 2 groups of arteries penetrate the surface of the spinal cord. There's vertical arteries and radial arteries. A series of vertical arteries arise from the ventral spinal artery, like the teeth of a comb, and pass in the ventral fissure towards the center of the spinal cord. They supply most of the gray matter and reach peripherally into the white matter also. In man, each vertical artery passes either to the right or to the left, supplying a territory on either side of midline. The radial arteries arise from all the other arteries on the surface of the cord. They supply the white matter and the outer regions of the gray matter. The spinal cord, therefore, has three vascular zones. The intervascular zone is supplied solely by the vertical arteries and the ventral spinal artery. The middle vascular zone is supplied by both the vertical arteries of the ventral spinal artery and the radial arteries of all other surface arteries. The outer vascular zone is supplied by the radial arteries alone. The problem of pulsation. It appears that large pulsating arteries cannot be tolerated below the surface of the neuroaxis, since the fluid nature of the brain tissue renders it incompressible. Large pulsating arteries would generate pressure waves below the surface. This may be why no large arteries travel below the surface of the brain or spinal cord. Arterial anastomoses of the neuroaxis. On the surface of the neuroaxis. Profuse anastomoses on the surface of the brain. These can sometimes provide effective alternative channels around an obstruction. The similar anastomoses on the surface of the spinal cord are finer and generally inadequate as alternative pathways below the surface of the neuroaxis. The deep arteries rarely anastomose above the capillary level. Some workers claim that there are no anastomoses at all above the capillary level. Others believe in occasional anastomoses at arteriolar level. Functionally, it matters little since these arteries below the surface are all functional end arteries. Thus, total obstruction of an artery below the surface provides death to all the neurons in its territory within about 8 minutes. And the arteries of the brain and the spinal cord may be blocked by a small thrombus or emboli. Such obstruction results in irreparable. I'm too tired for this. Irreversible damage. Let's leave it at that.